Hello biology students, and welcome to video 1 on our new unit, which is all about genetics. So what is genetics? Well, genetics is just the study of heredity, or it's the study of how traits are passed down from parents to offspring. So an example question you might look at when studying genetics might be, if your parents have brown hair, how is it possible for you to have blonde hair? We already know from our studies earlier in the year that all of our genetic information is carried on DNA and in genes that are located on chromosomes. So what we're going to be looking at in this unit is how are chromosomes passed from parents to offspring, and how do you determine what traits you have. So the biggest name in genetics is Gregor Mendel, or Mendel, and he is considered to be the father of genetics. He was a monk that lived in the mid-1800s, and he basically, just through careful experimentation and observation, figured out all of the basic rules of genetics and inheritance that we still study today. And I think one of the absolute coolest thing about Gregor Mendel is he was doing all this work in the 1800s. At this point, scientists didn't even know DNA existed. They didn't know chromosomes existed. They didn't know about alleles, different versions of genes. So he was able to figure out, how one allele is passed down from one generation to the next without even knowing that DNA and chromosomes existed, which is pretty impressive. And he figured this all out by studying pea plants that he planted in the garden of the monastery where he lived. So we're kind of lucky that Gregor Mendel chose to use pea plants because they actually work out really well for genetics experiments. There are a few reasons for this. So first, they work really well for setting up controlled experiments. Pea plants have both male and female reproductive organs, which means that they could fertilize themselves. The pollen, which is really plant sperm, from one plant can be used to fertilize the same plant, which is very useful for genetics. And they can also be used to cross-fertilize other plants. I could take the pollen of one plant and use it to fertilize a different plant to see what the offspring look like. Pea plants also show sharply contrasting traits, which means that if we looked at any trait, so for example, if we looked at flower color, the different versions of that trait, the different alleles, we've seen that word before, are very distinct. If flower color is my trait, the different alleles are purple and white, and in pea plants, they're either purple or white. There's no in between, so it makes it really, really easy to study the inheritance patterns. So in his experiments, what Mendel did, the first thing he did was identify what we call true breeding plants. And true breeding plants are plants that if you self-fertilize them, they'll always produce offspring with the same allele that they had when they started. So in other words, if I have a pea plant with white flowers, and I take the pollen from that pea plant and fertilize itself, all of its offspring will be white. It's true breeding. The same thing, if I had a pea plant with purple flowers and I took pollen from that plant and self-fertilized it, all the offspring would have purple flowers. We call this true breeding. So the first thing he did after he got his true breeding plants is he took two different true breeding plants. So he took a true breeding white pea plant and a true breeding purple pea plant and crossed them to see what the offspring would look like. Because he knew if he crossed the white plant with itself, it would give white offspring, and he knew if he crossed the purple plant with itself, it would give purple offspring, but he wanted to see what happened if they fertilized each other. And just for clarification, because these two plants are going to be the parents of the offspring plants, we call them the parental generation, or the P generation. The P generation is just defined as the first two individuals that mate in any genetics experiment. So when Mendel took his true breeding purple plant and his true breeding white plant and crossed them in that P generation, he noticed that all of the offspring were all purple. So he took a purple plant, he crossed it with a white plant, and all of the offspring ended up being purple. There was no white offspring plants. So these offspring the offspring resulting from this cross we call the first filial generation, or F1 generation. This is basically the first generation of offspring. We can also call them hybrids because they resulted from two varieties. I crossed a white and a purple. I crossed two different plants to get these offspring, so we can call those hybrids. The next thing Mendel did was take two of his F1 plants. So he took, he first started with the purple and crossed it with the white and got a bunch of purple plants. And he took two of these purple plants and crossed them together. So he took two F1 generation plants and allowed them to fertilize one another and got our F2 or second generation, F2 generation of offspring. And something even funkier happened because now 3/4 of the offspring were purple and 1/4 were white. And he was bamboozled by this. He didn't know what was going on. This was very strange because now this white trait that seemingly had disappeared popped back into existence. So kind of to summarize what's happened, because we're going a little quick, Mendel started with his parental generation, which was true breeding, which means if I crossed this purple flower to itself, the offspring would be all purple. Or if he crossed this white flower with itself, the offspring would be all white. But he took these two plants and crossed them together and got the F1 generation, which was all purple. He then allowed this purple plant to fertilize with another purple plant from the F1 generation, and all of the offspring from these crosses in the F2 generation were 3/4 purple and 1/4 white. By the way, here's another vocabulary word for you. Whenever we're doing crosses like this and we're only looking at one trait, we call this a monohybrid cross. Mono meaning one. So in this case, we're only looking at flower color, so we call it a monohybrid cross. Okay. So after looking at this data, Mendel made a couple of hypotheses. He first said, okay, clearly there has to be different versions of genes, because sometimes I'm getting white flowers and sometimes I'm getting purple flowers. So we now call these alternate versions of a genes alleles. We've seen that word a bunch of times in the last couple of weeks. So that was his first conclusion. And the second conclusion, which was really the most revolutionary conclusion that Mendel was able to make, was that every individual actually has two copies of each allele, or two copies of each gene. Which we know this because after looking at our meiosis unit, we know that chromosomes come in pairs, which we call homologous pairs. And Mendel realized for each gene, an organism must get one allele from each parent. Just like we know that egg and sperm cells are haploid and give one copy of each homologous chromosome to the zygote, to the offspring. So maybe his true breeding plants, like the purple plant, must have had two copies of the purple allele, and the white plant must have had two copies of the white allele. And then somehow when he crossed them, the next generation had one purple and one white. So depending on what combination of alleles you inherited, there are different vocabulary words that we can use to describe different individuals. So if you have two of the same alleles, so if you were a true breeding plant that had like two purple alleles, we would use the word homozygous to describe you. That means you have two identical alleles, and we could call that individual homozygote. That's the noun. If you had two different alleles, so say you had a plant had one copy of the purple allele and one copy of the white allele, we would call that individual heterozygous, or we call them a heterozygote or a hybrid. So remember, in the first cross Mendel did, he took a purple plant, which I'm now going to draw as two P's, because remember we have two copies of each allele, and he crossed it with a white plant that I'm going to draw as WW, and the offspring, because this was the P generation, and we know that the offspring, which was the F1 generation, this parent over here must have donated a P to the offspring, a purple allele, and this parent must have donated a W. But we know this F1 generation was all purple. So we have to have a way to differentiate between the purple and the white allele, because clearly they're acting differently. So in this case, the purple allele we call the dominant allele, because it's dominating over the white allele. Even though this W here is present, this generation is all purple. So we say that this allele is dominant. If it's present, it determines the organism's appearance. So the dominant trait will be visible if one of the homologous chromosomes carries it. And because it's dominant, we represent it by an uppercase letter. So for flower color, we're going to designate purple as being a capital P. So conversely, the white allele was being masked by the purple allele. So we're going to call this masked allele a recessive allele. It's the one that's hidden or masked by the dominant trait. So it's only visible when both homologous chromosomes carry this information. So if an organism had one P and one W, this plant would have purple flowers. It doesn't matter that this W is present, it gets masked or dominated by the P. So this trait is only going to be visible if the individual has WW, has two copies of that recessive allele. So I've been drawing it as a W just so it was clear to you guys, but in reality, we actually use the same letter as the dominant allele, we just write it as a lowercase. So from now on, my white allele, I'm going to write as a little p, which unfortunately looks very similar to my capital P with my stylus here. But the white flowers, or any recessive allele, we draw as a lowercase letter, the same letter as our dominant allele. So a couple of more vocabulary words for you to round out the video. So now we can look at the genetics of an organism at different levels. We can look at what type of alleles they have, and we can look at what physical traits they have, and they might be different. So the actual combination of alleles that an organism has, we call the genotype. So the genotype for a purple flower might be capital P, capital P. That means they have two dominant alleles. Another possible genotype for a purple flower might be capital P, lowercase p. If I had a white flower, their genotype would have to be little p, because we only see the recessive if we have two recessive alleles. We can look at an organism's traits is by looking at what they actually look like. And we call what you actually look like to be your phenotype. And kind of a good way to remember this, a little good word trick, is phenotype, which kind of sounds like it starts with F, even though it's PH, is the feature that you see. So for example, if an individual had a capital P, or capital P, lowercase p genotype, we would say they had a purple phenotype, because purple is the feature that you actually see. If an individual had a little p genotype, we would say that they have a white phenotype. So finally, let's do a little bit of practice. So what I'm going to do is I'm going to read the question out to you, and then after I finish reading the question, I recommend that you pause your video and try to answer these questions on your own before I give you the answers, because if you don't practice in your head, it will not be as useful for you educationally. So the example here is that true breeding tall plants are crossed with true breeding short plants, and as a result, the offspring are all tall. So go ahead, pause your video right now, and look at these 4 questions. Okay, everyone back? Good. Hopefully you paused and actually did that. So the first question is, so what plants are the P generation? And the answer you should have gotten is the true breeding tall plants and the true breeding short plants. Okay. So what plants are the F1? That's the offspring, the tall plants. That's the plants that resulted when we crossed the P generation. So now, what's the dominant allele? Well, the dominant allele would be tall, And we would use a capital T. And we know that's the dominant allele because if I took my true breeding tall plants that I'll write up here as capital T, because it's true breeding, so we can kind of assume it's homozygous. And we're crossing that with a true breeding short plant, which I'm going to write as little t, little t. So that means this parent's always going to donate a big T. This parent can only give a little t. So my F1 generation is heterozygous, but they're tall, which means that this capital T, the tall must be the dominant allele. And then finally, what's the recessive allele? That's the short allele. And we always represent recessive alleles as lowercase letters. So we would use the lowercase t. Okay, students, well, that's the end of the video, and we'll see you tomorrow.